So we just talked about what it means to be a macromolecule and what it means to be a monomer and a polymer. And now we're going to get right into the first macromolecule that we're going to talk about, which is carbohydrates. So carbohydrates, they serve as fuel and building material. That's the name of the section. Basically, carbohydrates are sugars and the polymers of sugars. So sugar would be the monomer, polymer is the bigger carbohydrates. And the simplest carbohydrates are what we call monosaccharides. Mono meaning one, saccharide is the suffix for sugars. And the macromolecules of carbohydrates are polysaccharides, which are polymers composed of many sugar building blocks. So poly meaning many, mono meaning one. Pretty straightforward. And monosaccharides, the monomers, they are usually multiples of CH₂O. So glucose is C6H12O6. And that's because of just the structure that it is. There's always going to be a couple extra hydrogens to fill in all those bonds that carbon has to make. Like glucose right there. And so we can classify monosaccharides by where the carbonyl is. Carbonyl is C double bond O, so you'll get really used to that. And where the number of carbons in the carbon, in the shape, in the skeleton. So that's how we can classify the monosaccharides that we're talking about. So here's a couple different ways. Here's some different monosaccharides. So here we have 3 carbon sugars, trioses. We can see that here the carbonyl is at the end of the carbon chain. So that's different than this one right here, which has it in the middle. We move on to 5 carbon sugars, and again at the end versus at the middle. And down here, our hexoses are 6 carbon sugars. There's some carbonyls at the end and carbonyls in the middle. And you'll actually see, this might come up later in the PowerPoint, but these 2 guys right here, they have a carbonyl at the end. However, they're flipped right here. They are enantiomers of each other, mirror images. So you can see how this one has the hydrogen on the left and the hydroxide on the right. Opposite over here, hydroxyl on the left and the hydrogen on the right. So those 2, that can make for so many different properties because we've just changed the ability of this molecule to interact with other molecules. That's what we do when we flip them like that. So that's why enantiomers are important. This is what we just went through. Okay, so we often draw the skeletons as straight lines like this right here. Up and down, it's easier. Carbon, 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 carbon. But usually they'll be in chains, or sometimes as they get larger and as they're in aqueous solution. So glucose is actually a 6-carbon ring. And that's how we'll draw it. We won't draw it in a straight line. We draw it as a 6-carbon ring because that's how it appears in nature. And basically these monosaccharides are fuel for cells and raw material for building sources. So those are the 2 main purposes of carbohydrates, fuel and structure. So here we see a straight carbohydrate. This is how we can write it, so you can like really see all the chemicals that are there. But once we see that it folds up and actually becomes a ring. And then we have disaccharides, which you could probably guess mean 2 monosaccharides bonded together through a dehydration reaction. So remember that one sugar loses an H and one sugar loses an OH. Those leave and become water, and now we've made a bond between the 2 of them. And this covalent bond is called glycosidic linkages. So you'll need to know the names of the bonds for all the 4 different four different macromolecules. So glucose or sugar is glycosidic linkages. So here we have, we're putting two glucoses together to make what's called a maltose. And so we see that this glucose is going to give up its OH right here. This glucose is going to give up its H. We lose a water, and now they're bonded together through a glycosidic linkage. Same thing down here. We're making sucrose, which is table sugar. So that's what you would like put in your tea or what you make cookies out of. And same thing, lose an OH, lose an H. Those leave as water through the dehydration synthesis, and now we have sucrose based on a glycosidic linkage. Then we get to polysaccharides, which are the polymers of sugar. And again, these have storage and structural roles. So we're going to use them to store energy and use them to build structures inside of living things. And the structure and function of a polysaccharide are determined by its sugar monomers and the positions of glycosidic linkages. So basically, which kind of sugar are the monomers for this polymer? So is it glucose? Is it fructose? What is it? And how are they bonded together? So the first one that we're going to talk about is starch, which is what plants use as fuel. How do they store their fuel? And it's made of glucose stacked on glucose stacked on glucose because plants make glucose in photosynthesis, so they need a place to store it. They store it inside these starch granules inside the chloroplast because the chloroplast makes the starch. So you can see that it's stored inside these big buckets of starch. And the simplest form of starch is amylose. So there's a couple different kinds of starches. This one is amylose. You can see that it's arranged all in a chain. Whereas up here, amylopectin is a different form of starch, and it's the same as amylose, except this carbon right here, they're branched. They're not in a straight line anymore. So this carbon is now bonded to 2, and it's going off in 2 different directions. So that's what we talk about when we talk about the structure of the carbon chain, whether or not it's in a straight line or if it branches off like this. Then animals use what's called glycogen, so it's different than starch. We store our extra glucose as glycogen, and we store them mainly in the liver and in the muscle cells. So here we can see this is mitochondria, and we can see some glycogen granules over there. So glycogen looks very different than glucose or amylopectin. It's very, very branched. So different than the amylose or the amylopectin that's found in starch in plants. It's very branched. And the purpose of these is that we use this sugar for energy, but sometimes, especially in plants, they're making so much they don't need it all right then. So they store their extra energy in these either starch or we store it in glycogen. That way we can use the energy later. And these, I don't know if there's an image of this. So, let's go back. Okay, so then a different polysaccharide is called cellulose, and this is what plants use to make their cell walls and make them be able to stand up straight. And so it's found in all cell walls, and it's a polymer of glucose, but the glycosidic linkages are different. So instead of the way amylose is structured, the glycosidic linkages that bond the glucoses together are different, and that make it better for structure than for storage. Remember, structure is always fitting function. So the structure of cellulose is more designed to be a structural molecule, whereas the structure of amylose is more designed to easily store energy for plants. And the difference between amylose sugar and cellulose and starch is because there's 2 different conformations of glucose, alpha and beta. And here we have alpha glucose. You can see that there's a hydroxide right here, and the hydroxides are right next to each other, so they're cis hydroxides. Whereas on the beta glucose, we have trans now. They're not right next to each other anymore. So it's a different conformation, and it makes for different bonds. So right here in starch, we count carbons based on their numbers, like which sugar number it is. So we start here, 1, 2, 3, 4, 5, 6. They have 6, right? Or I guess they have 5. Sorry, 5, because there's an oxygen right there. So over here, we're bonding the 1 carbon to the 4 carbon of the next one. This one's 1 carbon bonded to 4 carbon, 1 carbon bonded to 4 carbon in starch. Whereas in cellulose, we're still bonding 1 carbon to 4 carbon, but now it makes it upside down. So here's one molecule right side up, and then the next one is the reverse. The next one is the reverse, the next one is the reverse, just based on the fact that this hydroxyl group is switched from this one. So beta glucose leads to cellulose, which has been like glucose, 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 glucose. Whereas starch is glucose, 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 glucose. So they're flipped next to each other in cellulose. So polymers that have alpha glucose are helical, and polymers that have beta glucose are straight. So remember that cellulose has beta glucose, and if we flip all the way back here, you can see that amylose is twisted. So starch is twisted because it has alpha glucose, but it's really important that cellulose has beta glucose because they are used in plant cell walls. And basically what happens is we have a straight cellulose molecule, another straight cellulose molecule next to it. Now these 2 cellulose molecules can make hydrogen bonds with each other, and so now they're going to be tougher, harder to break, have more support because they can lean on each other. So that's really important. You can't do that with starch because it's twisted, but with cellulose, since it's straight up and down, we can do that. And so that's why it's such a strong molecule. And when a bunch of them are together and bonded with hydrogen bonds, we call them microfibrils, and this is what makes a really strong storage, really strong building material for plants. So you can see down here we have beta glucose, beta glucose, beta glucose. they're going like this, like I told you, and they're completely parallel, completely straight next to the next one. So we have this OH right here bonded to this. See, oxygen to hydrogen. Hydrogen bonds are always oxygen to hydrogen, and so it's just providing extra support. That way we have a bunch of microfibrils in a plant cell wall, which makes for the tough plant cell wall, which is why plants can even stand up straight. But it does make a little bit of a problem. So remember I said that even flipping that one OH changes a lot of things. So now the enzymes that we have that break down alpha glucose, they can't break down the beta linkages in cellulose. So when we eat plant material that has a lot of cellulose in it, we don't digest it. It just moves straight through our digestive tract as what's called insoluble fiber. And so how this works is it kind of irritates the inside of your digestive tract, and then your digestive tract produces a lot more mucus because it's being irritated, and that's its response. And so then, because of all its mucus, food moves through your digestive tract more easily. So that's how fiber works. Some microbes use enzymes to digest cellulose. So some microbes have special enzymes that can digest cellulose. And then these enzymes are often found in the stomachs of animals. Like herbivores, cows and termites, they eat only plants all day long. Cows eat so much grass, and they wouldn't be able to get really very much storage or energy out of that grass if they couldn't break down the cellulose, because they need to get to those glucose molecules, but for us, it's just passing right through us. We're not using energy from the glucose that's stored as cellulose. But these cows and termites, they have a symbiotic relationship, which is something we will talk about in great depth later, but they have evolved this crazy relationship with these microbes. So the microbes live inside their intestinal tract, and they break down the cellulose for them into the monomers, which is glucose, that the cows and the termites can then get energy from. And then chitin is another structural polysaccharide, and it's found in a couple of places, such as the exoskeleton of arthropods. So this guy right here, whatever strange insect he is, he's shedding his outer shell, and this would be tough based on entirely chitin, as well as it provides a structural support for fungi, so like mushrooms and stuff like that. They have chitin in their cell walls, which actually leads to evolutionary relationship. We'll talk about that when we get to evolution. But here's the structure of chitin. It has an extra group with a nitrogen over here, which is why it's a little bit different. But it's very, very strong, which you think about it, arthropods, like a lobster or any sort of shellfish, they're all made of chitin, so it's really tough. Think about their cell walls. Their exoskeletons. And so we can also use it to make a strong and flexible surgical thread that decomposes after wound or incision heals. So that way you can, it's super tough, but it'll break down after a wound has healed. That way you don't have to get your stitches removed. And that's the end of carbohydrates.